Hi and welcome back. Today we're going to be speaking about deep neural networks in brains and machines. This is the hierarchical connectivity structure of the monkey cortex. On the left you can see the hierarchical level, and here you can see lots of different brain regions with each line between the regions signifying a connection that exists in the brain. Here is an artificial neural network from the classic overview piece by Yann LeCun, Yoshua Bengio and Geoff Hinton. So our question today is just how similar are deep neural networks in brains and machines? By the end of the video, you'll be able to describe major types of neurons in the brain. You'll be able to describe how distinct features of the brain's anatomy and physiology change along a deep hierarchy of brain areas. You'll be able to sketch the basic architecture of deep and convolutional neural networks, and to critically compare the similarities and differences of deep and convolutional neural networks in brains and machines. So over 100 years ago, the modern study of neuroscience was really founded by a Spanish scientist called Santiago Ramón y Cajal. And Ramón y Cajal had not just an incredible scientific mind and an ability to look at details of neuroanatomy and imagine somehow very accurately the function of what he was seeing. He also had a lot of artistic talent and originally wanted to be an artist or a photographer. And you can see here some beautiful sketches he did of some of the first neurons ever seen under the microscope. And what you can notice is there are these central thick parts, which is where the cell body is. And there are also long protrusions from that central part, and these tend to be dendrites where information comes in. There are also some thinner outputs marked with a small a on these images, which is the axon. So there's quite a lot going on in this beautiful picture on the left, and here is a simplified version from Wikipedia. And what we see is these still major sections of the dendrites, which is where most of the inputs come in, the cell body, also known as the soma, and the axon, which is where the outputs from the cell tend to come from. And we can see that these dendrites tend to come and divide themselves into lots of different little branches. But what do we know about the computational capabilities of single neurons? Well, the Greek computational neuroscientist Panayiotis Poirasi investigated this question way back in 2003. And Poirasi and colleagues reasoned that now we're able to simulate quite detailed and accurate models of individual neurons, but it's quite hard to interpret those and understand what the key computations in these neuron models are. So their approach was to essentially do a model of the model and try to understand this complex, biologically realistic neuron model with a simplified neural network type model. They then compared multiple different neural network designs, including very simple ones where there was just a single unit, and they tested which of the models could best predict the response of the extremely complex and accurate model to multiple different types of stimulus input. And what they found was that a two-layer neural network with sigmoid nonlinearities was in fact able to predict very well the response of the complex biologically realistic model to many different types of stimuli. And in fact they went further and showed that these two layers were equivalent to a first layer, summing up all of the inputs on an individual dendritic branch and then passing that through a sigmoid type nonlinearity. And then all of those dendritic branch inputs are summed up at the soma, where it goes through another sigmoid type nonlinearity. So therefore they suggested that a biological neuron may be computationally similar to a two-layer artificial neural network. Where this was just one neuron, one pyramidal cell, which is a classic type of cell that's abundant in the cortex. However, not all neurons are the same. In fact, there's a principal division of neurons in the brain between excitatory neurons and inhibitory neurons. So an excitatory neuron is one that releases glutamate as its neurotransmitter. And when this connects via synapse to another neuron, which we call the postsynaptic neuron, this makes this postsynaptic neuron more likely to spike. However, inhibitory neurons release GABA as their neurotransmitter, and when that connects to a postsynaptic neuron via synapse, this makes the postsynaptic neuron less likely to spike. Now, when we say a neuron is glutamatergic or GABAergic, we mean that the main neurotransmitter that it emits is glutamate or GABA. And we can do this because of something called Dale's law. And that Dale's law says that a neuron releases the same neurotransmitters to all of its outgoing synapses. So if this neuron releases glutamate to this one cell, but it connects to multiple cells, then it will release glutamate to all of those cells. And this is approximately true, but like almost everything in biology, there are some exceptions. Within these categories of excitatory and inhibitory cells, there are actually many different subtypes. So here we see a beautiful illustration of the many different types of inhibitory neuron in a single area of the mouse brain. You can see different colors, the different cell types, and you see many different patterns of dendrites and axons. We can also see that if we look across brain areas, there are big differences in the excitatory cells. Here we can see an increase in the size of the dendrites as you go up the cortical hierarchy from V1, which is the first visual area, up to TEO, which is quite a high area involved in object processing. So we've seen in the first slide that the sensory cortex, at least in the primate brain, has this deep hierarchical structure. So what does that mean? What actually changes in the brain as you step up these hierarchy levels? Well, the first thing, and perhaps the most important thing, is that the time for a neuron to respond to a stimulus increases. So here we can see a standard hierarchical path of visual information through the brain. And we can see that information moves quite quickly from the retina to an area called the LGN. The LGN is a subpart of the thalamus, and this is the first stop inside the brain of visual information. This sends this information to the primary visual cortex, which responds after about 40 or 50 milliseconds. And this gradually takes steps further and further into the cortex before reaching the prefrontal cortex somewhere between 100 and 200 milliseconds. These times are actually a little bit longer for the human brain, and this is for the monkey brain shown here. So in addition to the timing of a response to a visual stimulus, also the types of stimuli that will cause a response change as you move up the hierarchy. So there are two important concepts here. One is the receptive field, and the other is the feature. So the receptive field is the part of space that a neuron is receptive to, or the parts of space that can cause a firing response in a particular neuron. And space here is defined as the visual space. So this is defined by the cells in your retina. So the cells in your retina lay out a map of the visual space, and you can read out this map also from the visual cortex in the brain. And certain neurons will preferentially respond to certain parts of space. At the first steps of the visual hierarchy, the neurons respond only to a very small part of space. You can imagine that they're seeing the world through a pinhole. Then as you step up the hierarchy to the secondary visual area called V2, the size of the receptive field gets a bit bigger. These neurons respond to stimuli in a little bit bigger amount of the space on the retina. And as we continue to go up the cortical hierarchy, the receptive field size increases. And up the very top, if you show the correct feature to a neuron here, almost anywhere in the visual space, this neuron will respond by firing. Now, I hinted there that receptive field was not the only important thing. Also, the types of features that neurons respond to become more complex as you step up the visual hierarchy. In particular, this is called the ventral visual stream, which really is how the brain decodes what it's looking at. And what we see is that as you go up the steps of the hierarchy, certain neurons begin in V1 by caring about edges and lines. Then as you go up a little bit further, you start to see areas that are sensitive to shapes, eventually areas that are sensitive to objects, and in fact faces. And the remarkable thing is that neurons up around IT and an area in the human brain called the fusiform face area, they can respond to faces no matter where they are in visual space and at different orientations and angles. Here we see that the number of neurons per brain area goes down also as you move up the cortical hierarchy. And this goes down for two reasons. First, the density of neurons per gram of brain weight goes down quite a lot, by about between 2 and 5 times from primary visual cortex to the prefrontal cortex. But we also see the size of the brain areas goes down, so that actually if you look in area V1, primary visual cortex, there are about 300 million neurons there in the monkey brain, whereas you take a few steps forward to area TH, which is connected very strongly to the hippocampus, there are only about 3 million neurons there. So it scales down by about a factor of 100. So this suggests that sensory information is compressed as you go from the early stages to deep into the cortical hierarchy. Now, although the density and the total number of neurons goes down along this cortical hierarchy, the number of inputs that individual neurons receive seems to go up. So we saw earlier this graph of the increased size of the dendrites of these excitatory cells as you go up the cortical hierarchy. And in particular, these are a certain type of excitatory cell called the pyramidal cell, which is by far the most common in the cortex. And that's what we're going to be focusing on when we talk about excitatory cells in this course. So in addition to the size of the dendrites going up, the number of synapses per neuron also goes up. And this can be measured by looking at the number of these little tiny protrusions from the dendrite called dendritic spines. And these are where over 90% of the excitatory synapses occur for pyramidal neurons. As we go up the visual hierarchy, again from V1 to higher visual areas in the temporal lobe here, such as TEO, we see a huge increase in the number of spines. And in fact, as you go from the bottom of the hierarchy to the very top, even into prefrontal cortex and another nearby area called the anterior cingulate, you can see about a 10 times increase in the number of spines or the number of synapses along the cortical hierarchy. So in addition to this increased number of inputs, we see that there's this increased capacity for neuromodulation. And in particular, in our recent study, we found that as you go up the cortical hierarchy, shown here on the y-axis, the number of receptors per neuron goes up by about 4-fold. And a lot of these receptors are actually for neuromodulators such as dopamine, serotonin, acetylcholine, etc. So there seems to be an increased capacity for neuromodulation, so for quick, rapid changes of connectivity strengths, for example, as you go from the early sensory areas at the bottom of the hierarchy to the more higher cognitive areas, such as prefrontal cortex near the top of the hierarchy. So when we say the hierarchy or the cortical hierarchy, it might seem that there's just one ordering of everything in the cortex. And there are indeed some researchers that believe that. But I think a more conservative, but probably more accurate view is that there are in fact different hierarchies in the cortex, and these correspond at least to the different sensory modalities. So here in this absolute classic work by Fellman and Van Essen from 1991, we see a map of the visual hierarchy on the left and the somatosensory hierarchy on the right. And the somatosensory system is the system that processes touch. There are hierarchies for auditory information and perhaps a little bit less well studied, but also the other senses too. Now, one important part of this hierarchical structure of the cortex is the feedback connections. Although we've talked about information propagating through the system from going from the early sensory areas up to the cognitive areas, in fact there are just as many and perhaps even more connections that go in the opposite direction from areas like prefrontal cortex back down to the sensory areas. Here we can see some of the functions of feedback connections, and this includes selective attention. And there are two types of selective attention. One is bottom up, which is stimulus driven, which is when a particular type of stimulus is salient, it grabs your attention and you lead your focus onto that. Another is top down or goal directed attention. And this is when you have a goal in mind and you want to focus on this particular thing to the detriment of some other things, but you might be able to process this information somewhat better. And here you can see a little diagram. The brains are facing to the right this time, so prefrontal cortex is over this side of the brain. Here we can see a little diagram of some important feedback connections showing how parts of the prefrontal cortex connect back to early sensory areas via intermediate areas that include higher visual areas and parts of the parietal cortex here. There are too many papers to cite on this, but there's a lot of beautiful work from Tyrone Moore's lab, and we're going to put in some references at the end. So it's thought that these feedback connections might make it easy to focus your attention on a particular feature or a particular part of the visual space, because they might excite a little bit neurons in the visual system that preferentially process that feature or that part of space. Now, this feedback connectivity from these higher areas is unlikely to directly cause these sensory neurons to spike, but it might excite them a little bit so that they're closer to the threshold for spiking, so that when a stimulus does come in, they can move and transmit information and spike quite easily. And it seems to have actually quite opposite effects on other features and other parts of space that you're not focusing your attention on. So in a way, if you could think that all of the information that's coming into your visual system is competing for access to your mind, to your attention, and eventually to your working memory, well, this top-down connections, this goal-directed top-down attention biases that competition so that certain features and certain parts of space are more likely to win that competition and become the focus of your mind. Another potential role of top-down connections is in transmitting predictions from higher parts of the cortex back down to earlier parts of the system. And this idea was proposed and beautifully described by Rajesh Rao and Ballard in 1999. And they suggested that there are two important streams of information from the top of the hierarchy back down. One step of the hierarchy come the predictions, and at this level, when you get new sensory information coming in, there's a comparison between this real data that came in and what was predicted, and this comparison then creates a prediction error, and this prediction error is passed back up to the next level. And this predictive coding phenomenon, as it's called, is now a very popular, and I would say one of the leading theories in neuroscience. And it has some experimental evidence behind it, but I think there's a little bit left to go to say that it's proven. More generally, apart from selective attention and predictions, these top-down connections, also known as feedback connections, are essentially a means of providing some sort of context for ongoing sensory information and how you process that. So I've talked a lot about cortical hierarchies and in particular about hierarchies in the visual system. And what this seems across many of the different measurements that we were looking at, from physiology, anatomy, etc., to be really quite clear, particularly in the visual system. And here you can see a calculation of one measure of the hierarchy, and as you go from V1 up through a few steps of different sensory areas, you can see this really clear increase in this hierarchy value. I won't go into the details of this, but this is dependent on how different brain areas connect. However, once you get from these sensory areas into areas that are more strongly involved in cognition, it becomes much more difficult to separate these areas in terms of their hierarchical value. And in fact, there's an ongoing debate about whether there is actually a hierarchy at all amongst these different cognitive areas. So I would say that these hierarchical neural network structures certainly seem to be accurate for individual sensory systems and a very good approximation, particularly if you include the role of feedback connections. However, it's a little bit more muddy and unclear whether such a hierarchical system, such as a deep neural network, is in fact a good model of higher cognitive systems in the brain. So now we're going to turn from the brain to artificial neural networks. And we saw what a biological neuron looked like, and now we're going to look at what an artificial neuron, or a unit in a neural network, looks like. Now, one of the first types of artificial neural networks was proposed by McCullough and Pitts back in 1943. And this was quite simple. And what it said was that if you were given a list of multiple different inputs, X, the first step was to sum all of those inputs. And these were binary inputs of ones and zeros. Then, if this sum was greater than a threshold theta, then you would output a 1, and if it was below that threshold theta, you would output a 0. This was replaced in the 1950s by Rosenblatt and others by a sigmoid, and the sigmoid function again maps between 0 and 1, but it does this much more smoothly, so that if there is a threshold, it's somewhere soft. You can see the sigmoid function here on the right. Now, more recently, people have started to use many different functions. These are known as activation functions, in order to convert these summed inputs into an output. And these nonlinearities are very important for giving computational power to the network. And one of the most popular nonlinearities now, or activation functions, is called the ReLU. And you can see this in blue here. The ReLU works as follows. If you sum up all of the inputs from the different weights and a bias term if there is one, and if all of that sum is a negative number, well, you set the value of the output to 0. And if all of that sum is not a negative number, then you just leave it as it is. So it's called a rectified linear unit. So here we see one of the classic problems in machine learning. And it's a classic problem because the first stage of neural networks, with just a single layer, we're not able to solve this problem. And this is a problem that requires finding a nonlinear boundary. And a nonlinear boundary is something that is not a straight line. So here, for example, we want the network to respond with a 1 if x2 is plus 1 and x1 is minus 1. And we want to respond with a 1 if x1 is equal to 1 and x2 is equal to minus 1. But we want it to respond with a 0 if both x1 and x2 are equal to plus 1, or they're both equal to minus 1. So this is called XOR, or exclusive OR, because you want the network to respond if x1 or x2, but not both, are active with a plus 1 value. This reason a linear system cannot solve this is because there's no single straight line in this space that can separate the orange dots from the green dots. However, if you introduce depth in the form of at least one hidden layer, you can solve the XOR problem. So this essentially continues the logic from the previous step. You have some inputs. You have these inputs map on to some units in a hidden layer with weights W1. These W1s are multiplied by the strength of the inputs. And this is added up and added to a bias term if there is one. And this is put through one of the activation functions or nonlinearities that we saw on the last slide. This is then propagated on to the following layer, which could be a further hidden layer or already the output layer. Again, this is done with a different set of weights called W2 here, and this propagation from the hidden layer to the output layer is again done by summing up all of the hidden layer activations according to the weights of their inputs to the neurons in this output layer, plus a bias term if there is one, and then putting it through the nonlinear activation function here. This can then be converted into a decision, such as whether there was a cat in the input. So the important thing to take home from this is that depth and nonlinearities can help networks solve complex classification problems that simple linear networks or shallow nonlinear networks are not able to solve. And the key idea here is that the output y is not computed directly from x, but passes through another set of weights, or maybe multiple other sets of weights, via nonlinear activation functions as well. And now we can find nonlinear separation boundaries between different categories in a set of data. So one of the big problems for deep neural networks was getting them to realize that a dog is still a dog and it's the same thing no matter where it is on the screen. So in the brain, there are certain neurons that will respond to dogs no matter where they are, no matter which direction they're looking, how they're lit, etc. This is called translation invariance, as the response of this neuron in the brain won't vary very much if you move the dog around the space. However, neurons in feed-forward networks that are fully connected, such as the ones we looked at so far, they do not tend to have translation invariance emerge naturally, unless you show perhaps all of the possible positions that you will see a dog in. You'll end up getting some neurons that specialize for dogs in particular locations and others that specialize for dogs in other locations. So how does the brain actually solve this problem, and how does the brain produce neurons that are sensitive to particular features that are invariant to translations of that feature around the visual space? Well, here we can see an idea of two types of cells in the visual cortex. The first is what's called a simple cell, and this is similar to what we mentioned earlier. So a simple cell will respond to an image if the image contains the feature that it is interested in, if the feature is in a particular part of the visual space that corresponds to its receptive field. And this is shown here in white. Interestingly, there are parts bordering the receptive field that neuron is likely to be inhibited by. So the response to this biological neuron is largely predictable by how much it likes this feature and how well it overlaps with its receptive field. But the biological neurons tend to also impart some sort of a thresholding function, so that if this neuron is not sufficiently activated, perhaps not sufficiently overlapping with its receptive field, it will have very little response, or perhaps none at all. But once you pass that threshold, it will start to see quite a big response. These responses of these simple cells are then added up in the next stage of hierarchical processing, which you see in something called a complex cell. And what complex cells do is they sum up the individual responses of many of these different simple cells. And these simple cells will be sensitive to the same feature. It could be, for example, a line going up this direction here. But they're sensitive to that same feature in different parts of visual space. So what we see is that these complex cells become sensitive to the same feature as all the simple cells that lead into it, but now they respond to this same feature over all parts of the space. So very much with this in mind, Yann LeCun and colleagues invented convolutional neural networks. So as a reminder, convolutional neural networks are very important for the recent history of AI, as in 2012, the modern resurgence of interest in AI really kicked off with the incredible performance of the AlexNet convolutional neural network on the ImageNet challenge. And this has really led to the last more than a decade of research in this area and funding and investment, and to more recent developments that are very exciting too. So there's a very clear analogy between the way a convolutional neural network works and what is known about certain parts of the visual system in humans. So much like neurons in the visual system, convolutional neural networks work such that each neuron in a particular layer of the system has both a receptive field and a preferred feature. The receptive field works as follows, that this neuron here will just pay attention to a particular patch of the image, and it will like a particular feature. And these features in this example will be a gradient from white to grey to black, or from high contrast to medium and low contrast. And what the convolutional neural network will do is that for this particular neuron, it will look for this feature in this patch of the image. We can do this essentially by doing an element-wise multiplication of this feature window here with this receptive field on the image here. This first column of the element-wise multiplication is 3 times 1 plus 1 times 1 plus 2 times 1, which equals to 6. The second column is all zeros, and the third column is minus 1 plus minus 1 times 8, that's minus 9, plus minus 1 times 2, so that's a total of minus 11. So if we add up these three things, we will get 6 minus 11, which should be minus 5. There we go. So the next neuron over uses exactly the same feature, but now looks for this feature in the next part of space over. And if we can do this again, this column is going to be equal to 12, this equal to 0, and this equal to minus 16. So there should be a minus 4. And we can continue this trick using this convolutions to search for the same feature in each part of the space of this image. So in practice, after performing this convolution, you would then pass this intermediate result through a nonlinear activation function such as the ReLU. Following the convolution and the activation function, there is commonly a pooling operation which is done partially to reduce the size of the network and reduce the number of parameters. And this is commonly a max pool operation. And the way this works is by dividing the image into different sections and taking just the maximum value within that section. Now this results in a reduction in the number of units or number of neurons in the model as you get deeper into the model. And this seems to have some resemblance, at least on a superficial level, with the decreasing number of neurons that you get along the cortical hierarchy in the primate brain, at least. Sometimes people also do a normalization of the activations, for example, within a layer. This is actually very similar to something that is seen in real neurons, that very often the activity of a neuron is essentially divided in practice by the activity of nearby surrounding neurons. And this seems to accentuate the differences in firing between neurons. So this is essentially what a full convolutional neural network may look like, with multiple different convolution, pooling and normalization steps. It often ends also with fully connected layer or two before some sort of a classification. So to recap what we talked about today, biological neurons are capable of more computation than artificial units or neurons in neural networks. They seem to be equivalent to at least a two-layer neural network. But biological neurons are themselves very diverse, with many different subtypes of excitatory and inhibitory neuron, as well as neuromodulatory neurons that release other transmitters such as dopamine, serotonin, etc. We saw that there are different sensory hierarchies in the cortex, and we focused in particular on the visual hierarchy as one very good example. And we also saw that as you move up these hierarchies in the cortex from the early sensory regions to higher areas, the response time of a neuron to fire to sensory input increases. The receptive field gets bigger. The preferred stimulus gets more complex. The number of neurons from area to area goes down, suggesting some sort of a reduction in the dimensionality of the representation. The number of inputs per individual neuron goes up, with perhaps 10 times as many synaptic inputs on individual neurons in prefrontal cortex compared to primary visual cortex. The capacity for modulation goes up as well, with many times more receptors for neuromodulators in prefrontal cortex than primary visual cortex. We saw that it's debated whether there is in fact a hierarchy amongst different areas in the higher cognitive parts of the brain. We saw, moving to machine learning, that depth and nonlinearities allow neural networks to solve complex classification problems. And we learned that convolutional neural networks are in fact directly inspired by features of biological vision processing. That includes the hierarchical structure, the receptive fields, and the feature detectors. So thank you very much. If you want to learn more, I suggest you choose one of the papers on this list and enjoy. So although the next video lecture is actually about recurrent architectures in brains and machines, very soon following that we're going to learn about how to train these deep neural networks and learn about how they compare to learning in brains.